que puedan conocerlo. Y nuevamente, mil gracias por haber aceptado la invitación a nuestro ciclo de seminarios. Hola. Adelante, Ma.
Sí, pues muchas gracias. Buenos días a todos y gracias por estar aquí a las 11 en lugar de a las 12. Les va a gustar mucho el seminario del doctor Colin Bonduel. Él trabaja en la Universidad de Burdeos, en Francia, en el laboratorio de polímeros. Y bueno, les platico un poquito de los antecedentes de Colin. Él obtuvo un doctorado en la Universidad de Toulouse, en Francia, y estuvo trabajando, pues su tema son polímeros, ¿no? Entonces su tesis de doctorado fue sobre cómo sintetizar algunos poliésteres. Después estuvo 2 años haciendo un postdoc en la Universidad de Western University en Ontario, Canadá, haciendo algo que se llama grafting, que es decorar superficies con polímeros. Es un proceso que involucra diseño. Y bueno, como les decía, desde 2019, Colin ya está trabajando en la Universidad de Burdeos. Tienen unas instalaciones muy impresionantes. En marzo tuve oportunidad de visitarlo y pues trabajan allá pues ya ni siquiera en grupos, ¿no? Son grandes consorcios de una decena de investigadores al menos, súper equipado. Y bueno, Colin entonces es experto en polimerización y tiene una rama en su investigación que involucra, como él ya les va a platicar, tratar de hacer proteínas artificiales con este tipo de polimerización que él maneja. Entonces, bueno, pues le cedo la palabra a Colin. Colin habla muy bien español. Ha decidido darnos su seminario en inglés, pero la discusión, las preguntas, sin ningún problema, pues las pueden hacer en español o en inglés. Entonces, adelante, Colin, y muchísimas gracias por aceptar la invitación. Bienvenido.
Sí, muchas gracias, Marcela. Muchas gracias por esta invitación. Sí, es cierto que puedo hablar o intentar hablar en español, pero sí es más fácil para mí como con el vocabulario y todo hacerlo en inglés. Y sí es cierto que de lo que vamos a hablar hoy es de cómo, digamos, pues voy a compartir, cómo digamos, de las proteínas podemos ir hasta, como se diga, a la química de los polímeros, ¿no? Ok. Y me falta, ok, perfecto. So, my name is Colin Bonduel. I'm from the Laboratoire de Chimie des Polymères Organiques from the University of Bordeaux. I'm working at the Seminario, so it's a national agency in France. And we are basically pure researcher, and we are mixed in several different laboratories. And we are mixed with a teacher, professor. And our goal is to strengthen the research in France on a specific topic. And mine is about using protein as natural model to develop some polymer chemistry. And that will be what I will develop today. So of course, I will not explain to you what are proteins, how fascinating are these macromolecules for living system for us, etc., etc. I think you know that the protein, they have a specific sequence of amino acid, which is very important. And thanks to this specific sequence, you have a local organization in secondary structure and a tertiary folding of the structure that is finally at the origin of all the essential functionalities of protein in living systems. I think what I would like to highlight today is that protein, they are also what we call natural polymers. So polymer means that finally they are macromolecules that consist in a repetition of the same monomer unit. And the same monomer unit is actually the amino acid unit. So you have the specific repetition of this unit with this chiral center here. You can have different lateral chain, but you have all this alpha peptide linkage going together and forming these polypeptides. So what is interesting to start with is to consider how peptide or protein can be useful. And in particular, they are currently developed in 3 big applications. So they are very useful in therapy, and you know that very well. For instance, as bioactive molecule, but also as drug delivery system. They are more and more used as cell matrix, so to develop, for instance, specific ink for bioprinting and so on. And also they are very used in cosmetology. And these are 3 key applications we are currently working on here in the laboratory. So as you may know also is the protein synthesis happen in cellulose, in living system, through the ribosomal protein synthesis. So you have this complex system, so protein mixed with nucleic acid that is able to template message RNA and to form like this sequence control macromolecule that we call proteins. And from this specific sequence, after some post-translational step, forms this specific structure of proteins. What is interesting here is to consider the use of chemistry to mimic this structure or to prepare proteins. And in that case, the first thing you can have in mind is the use of what we call the solid phase peptide synthesis. So solid phase peptide synthesis is this old method now that has been fully developed in the 60s, so more than 60 years ago, where you have several coupling steps in chemistry to fully reproduce the complex primary sequence of proteins. This has been has known a lot of progresses, and in particular now, since 2 decades, this is combined to what we call the chemoselective amide bond forming ligation, where you have 2 peptides that can be efficiently combined through a process that is named also native chemical ligation. So these 2 approaches are very developed. So far, you might face some significant limitation that I will present further, but there is also another methodology that we present less, particularly when we are in biochemistry or in biotechnology field, that is a polymerization process. So what polymerization process means? It means that here we will produce a polypeptide backbone in only 1 single step. And we don't have to use this complex iterative coupling of different amino acids. So this process is based on a molecule that is named N-carboxyanhydride that correspond formally to the condensation of a CO2 molecule to an amino acid. So it forms this N-carboxyanhydride ring that is activated and that will react very efficiently with a nucleophile that can be an amine, and that ring open this NCA moiety, make CO2 molecule removed from the mixture and regenerate an amine end extremity. And this amine end extremity can reattack a new monomer and so on through a propagation process. So what is interesting here is you mix basically NCA with small amount of amine, you tune the stoichiometry of your amine with the NCA, and you can define basically the size of your polypeptide. Even so, you don't really control the sequence of your polypeptide. And this is the first thing, is with this ring opening polymerization method, you can easily tune the size of your polypeptide in 1 single step. Also, you can play with what is called macromolecular engineering. So depending on the initiator you use, if you have multiple amine groups, you can have branch structure. You can play with specific initiator to have microcyclic structure or star-like structure and so on. So you have really a simple tuning of your system by well choosing your initiator and the stoichiometry you use for the size of your polypeptide. So let's go back a bit more in detail in the comparison of the solid phase peptide synthesis, so the iterative coupling, and the ring opening polymerization, so the simplified polymer I propose in this discussion. So solid phase peptide synthesis is what we call in chemistry a total synthesis approach, where the goal is really to reproduce the full structure of a protein. So let's say a protein has 200 different amino acids, sequence control. So what you will do is specifically all this sequence, you can use native chemical ligation to afford this convergent synthesis that can help. But at the end, you will be able to form oligomers. And in the best case scenario, you can reach the real size of a protein, but it's still a challenge in many cases. In the case of ring opening polymerization, we have a simplification of the system. So instead of to have a complex sequence of amino acids, we just use a mixture of amino acids. Somehow it can be just one single amino acid, and you make a homopolymer or copolymers. The advantage here is you make gram scale of your macromolecule of your polypeptide, and they can have big size, so size that are in the range of real protein. So simplified analog versus true total synthesis of protein. So the more fundamental question besides that, and that's something maybe we can discuss, it's very interesting, is to consider whether the sequence of a protein is always necessary to reproduce the structural or the functional feature of this macromolecule. So in biology, many of you strongly support the idea that everything relies on the primary sequence of protein. But actually, and it's more and more obvious, you can consider protein first as macromolecules that are made of amino acids. And beside this primary sequence, what can matter also in many properties, physicochemical properties, and even biological properties of protein is the size and the nature of the main backbone rather than the specificity of the primary sequence you can have in this macromolecule. And these are some keywords in which it's known now that even so you don't really control the primary sequence, you can have a good control and a good reproduction of the protein properties in physicochemistry. So conservation, co-formation, aggregation, and so on. So I will give you a precise example to support this. It's about the secondary structure of proteins. So as you may know, secondary structure, there's this conformation that are stabilized by hydrogen bonding network. So you can have an intramolecular hydrogen bonding network that form helices, and the main one that is known is the alpha helix. And you can have an intermolecular hydrogen bonding network that are the beta sheets. And actually, what is very interesting is even so you don't have a control in the primary sequence, with polypeptide polymer, you can have secondary structure. And this is explained by the fact that the lateral chain of the polypeptide is not at the origin of this secondary structure. The hydrogen bonding only involves so far the peptide linkage of the main backbone of the macromolecule or of the polymer, for instance. The side chain only influences this secondary structure. So, and that it's actually something known since a long time. And if you go back to the 50s, actually the work of Pauling and Corey, the first work in structural biology of proteins to decipher the structure of proteins, they have used this polymer to make the simple footprint on circular dichroism of an alpha helix and on of a beta sheet structure. So they have been this polymer a long time ago tool for structural biology, just because they were able to reproduce this secondary structure. Now, and what is even more interesting to me is that this secondary structure is a powerful chemical leverage to tune many properties, and in particular to promote polypeptide in advanced materials. So that can be the secondary structure can tune the hydrodynamic volume, for instance, of the block of the polypeptide, can tune the rigidity of the polypeptide, the viscosity, the microphase separation, the thermal stability, the macromolecular chemistry you can do with your polypeptide, and so on. So today, I wanted to challenge a bit 4 keywords coming from the protein world, I would like to say, And now we were able to take this keyword and to push the polymer chemistry to reproduce as much as possible what natural systems made of protein are able to do. So these keywords are the self-assembly. So protein systems, they are known to be very organized and through very specific rules that you can mimic somehow with these polymers. The adaptiveness of the protein system, the fact that the protein system, they are systems that are built in an aqueous medium, and also the fact that protein systems are bioactive. So let's start with a secondary structure, so the alpha helix. And considering that the secondary structure, for instance, the alpha helix, they have macrodipole, which is actually an addition of small dipole coming from the peptide linkage that are going in the same direction. And this macrodipole in proteins, they are strong driving force for their organization that can be folding or self-assembly. What is interesting here is because with polymer, we are able to reproduce this alpha helix, the ring opening polymerization is a simple methodology to vary in one single step the size of the helix. So basically, with the ring opening polymerization, you choose the value of your macrodipole to promote a self-organization process. And I will give you one example here that is not directly linked to a biological system, that is more linked to a material science application, where sometimes people, they want to make what we call composite, and in particular, nanocomposite. So it means that you mix an inorganic phase that are inorganic nanoparticles in a polymeric phase that can be, for instance, diblock copolymer. So without entering all the rules of diblock copolymers, they tend to self-organize and, for instance, form lamellar phase. And depending on the nature, the chemical structure of this diblock copolymer, and depending on the nanoparticle, you can basically localize the nanoparticle somewhere in one of the phase, and then pattern the nanoparticle and provide to your material specific properties. So here is the formulation people usually perform. It's nanoparticle and the diblock polymer. What we have tried in this work was to directly anchor a polymer on a new nanoparticle and use the alpha helix structure, so the macrodipole, to tune this lamellar phase formation, this self-organization. So we did the ring opening polymerization from this monomer. So it's basically a glutamic acid that is benzyl protected here, and it's forming this N-carboxy anhydride. So you can do the ring opening polymerization. We have used here the propargylamine to initiate the ring opening polymerization. So attack here, remove CO2, make here different lengths. So these are the polymerization degree from 28 to more than 400. And then you can mix new nanoparticles. So they are specific nanoparticles without oxidation at the surface. And so if you know coordination chemistry, you can have like coordination chemistry between this alkyne group at the surface of the nanoparticle. And then you anchor the polypeptides, and if you mix them, then you can have this nice self-organization of the nanoparticle, creating this new kind of nanocomposite. So what is good here, it's really easy to tune the size of the lamellar morphology by just tuning the size of the macrodipole. So basically by just tuning the polymerization degree of the polypeptide. And just by changing this polymerization degree, we can have patterning size from less than 10 nanometer up to 50 nanometer. So these have a lot of impact, and I will finish here for this first part. It's this size and this macrodipole can have a lot of influence, and in particular for piezoelectric application. And it's true. I will not enter in this in too much detail, but the alpha helix you can form from this polypeptide reproduce the piezoelectricity you can have in some crystal of real proteins. And it's on what we are currently working. So then I wanted to develop a second keyword, that is the adaptiveness, considering that in biology, protein, they are very complex systems that are able to adapt to their environment. So going back to polymer chemistry and to polypeptide chemistry here, it's known since a long time that simple homopolymer, for instance, poly-L-glutamic acid, repetition of glutamic acid here units, these homopolymer, they are stimuli responsive. What does that mean? It means that, for instance, if they are in aqueous solution here, let's say at pH 4, they form an alpha helix structure. This corresponds to the creation of an hydrogen bonding network that stabilizes this conformation. If you change the pH of the solution up to 7, then you deprotonate the side chain here, and you have negative charge that tend to repulse all the lateral chain and disrupt this hydrogen bonding network. And you pass from an alpha helix structuring to a coil disorder state. So basically, if we summarize this, poly-L-glutamic acid, they are stimuli-responsive polymers, and they are sensitive to pH changes. pH is a good trigger. Actually, here it's just an example of polyglutamic acid of 27 units with a circular dichroism of the alpha helix and the coil disorder state. So pH is a good trigger, but what we have worked on the last year was to adapt this trigger to be even more relevant to some class of proteins. First one could be the use of metal to induce some sort of structuring, and it's known. A lot of proteins in nature, they use metal, not only for catalysis, but also just for structuring. And zinc is a good example, for instance. And here it's a work where we have made class of polypeptide where metal coordination is able to promote the secondary structuring. And if you remove the metal by using, for instance, an external ligand, competitor to the coordination chemistry, you can retrieve the coil disorder state. So it's, in that case, a secondary structure, a stimuli response to metal coordination. Even more challenging was to work on DNA interaction. And it's known in biology that proteins, they are able to interact with nucleic acids. And this interaction can promote the formation of secondary structure. And this happened, for instance, with leucine zipper, where you have a part that is in coil disorder state that becomes alpha helical in contact with DNA. So here we have made this principle much more simpler by creating polymeric analog of peptide nucleic acid. So here is the full methodology toward these polypeptides. So first, we start from an amino acid that is protected here with a propargyl alcohol. We have made the N-carboxyanhydrides, and then we have done the ring opening polymerization using xylamine. So we open the stoichiometry of this xylamine as compared to the monomer, permit to control the size of the polypeptide. And then in the last step, we do Huisgen cycloaddition. So it's a famous click chemistry. And we have used a drug for that because drug makes this molecule cheap and you can easily use it to make materials. And when we have used this drug, it's because it permits to include a nucleobase that is a timidine. We put the nucleobase on the lateral chain of the polypeptide. So unfortunately, the polypeptide was not directly soluble in water, but if you premix this polypeptide with adenosine monophosphate in DMSO solution, upon dialysis, it becomes soluble, just because this base pairing permits to stabilize the system with this phosphate group. Then it was possible to measure the secondary structure of this nucleopolypeptides in water. So it's alpha helical, as you can see here, the 2 minimum. And so just to remind you, It's a polypeptide with thymidine here that are in interaction with adenosine. Then what is interesting here is you can mix this water solution with DNA sequences. And here I give you two examples. Basically, DNA replaces AMP because of the multivalent effect of the interaction of the base pairing. And we have tried a sequence where we just have oligo A sequence and a sequence where we put some C spacer between each A. And what you can see is the oligo A sequence really shift the secondary structure of this polypeptide towards something that is closer to a beta sheet structuring. We explain this roughly for the moment by considering that the beta sheet better match the distance required by an oligo A sequence for base pairing as compared to alpha helix. To get more solubility, we have even included a phosphate group directly onto the nucleobase before the Huisgen cycloaddition. And in that case, we directly got beta sheet structure polypeptide, but that were ionic and with unusual beta sheet stability. So this means that they are in beta sheet structuring, even so they are fully water soluble. So it's kind of a weird amyloid structure, fully water soluble, that interacts specifically to oligo A sequence. What is interesting in that case, by the fact that we use polymer chemistry, is you can really translate that in what I presented you in the introduction in macromolecular engineering. So it means that you push the system, for instance, in a die block architecture, and then you can promote self-assembly in nanomaterial that can be micelle, that can be vesicle, etc. But you have at the surface of this nanomaterial and a polypeptide that is able to recognize specifically oligo A sequence, which is usually something you achieve with much more complex molecules or macromolecules. Okay, this was the second example of adaptiveness, where we have tried to develop polymers that adapt pH changes, metal concentration changes, and finally, nucleic acid sequences. I will enter in a new keyword now, the aqueous system. Just I repeat this slide from the introduction where I presented you the ribosomal protein synthesis, the solid phase peptide synthesis, and the chemoselective amide forming ligation that we also call native chemical ligation. If you think about the chemistry as compared to biology, what you realize is in chemistry, we don't really use aqueous system to reproduce the structure of proteins, the structure of polypeptides. So it's what we have tried with our polymerization process. So this is an N-carboxyanhydride I already showed you for the nanocomposite, which is a glutamic acid derivative with this benzyl group, and you have the NCA moiety. The problem with the NCA moiety is sensitive to moisture. And basically, if you put the BLG NCA in water, what you expect is to lose this CO2 molecule because you have a water molecule that will attack here. And what you obtain at the end is just an amino acid. So water on the paper is not really a solvent you should use for this ring opening polymerization process. Even so, we realize that in specific aqueous conditions, here, for instance, with a carbonate buffer and at 4 degrees, here it's the size exclusion chromatography of what we obtain. We realize that not only amino acids were produced, but also oligomers and polymers. That we attributed to the fact that the amino acid we prepare from the water initiation of N-carboxyanhydride is able then to propagate and form some macromolecules. So this is interesting, but not very controlled. So it's a sort of complex mixture of polymers. And we don't really like that in polymer chemistry now. So what we have done is to really use this result and try to push it in a process that is more and more developed in polymer chemistry, that is a polymerization-induced self-assembly. So here it's how we did that. You have a polyethylene glycol. So with this amine end extremity. Here's the same size exclusion chromatography. So just to remind you, the size exclusion chromatography is a chromatography that separates the different size of a system with the refractive index. So you have a polydispersity that is quite low with polyethylene glycol. So it's almost monodispersed. And if we do the same ring opening polymerization in presence of polyethylene glycol, instead of to have a complex mixture of polymer, here in that case, we specifically propagate the ring opening polymerization at the end extremity of the polyethylene glycol. So here is how the reaction mixture look like before the ring opening polymerization, what we call ROPISA in that case. And this is after, so it's like almost 2 hours after. You have this opalescent solution. And if you go further in this solution, you realize that it's full of nanomaterial actually. And this is basically the driving force of this process. Formation of nanomaterial, phase separate the system, and is the driving force of the ring opening polymerization. Template this ring opening polymerization and avoid NCA from hydrolysis. So what is interesting is we have a process that is fully achieved in water. We can have different solid content, and we are very reproducible in terms of size of nanomaterial. So it's something we worked on a lot. We have a patent now. We are transferring the technology to a company here in France. And just to show you some work specifically on secondary structure, because I presented you that in the first slides. You can use in this system different monomers, so the same N-carboxyanhydride here, the BLG N-carboxyanhydride, but also you can use the leucine N-carboxyanhydride that will drive, instead of alpha helix secondary structure, a beta sheet secondary structure. As you can see here, this has a strong impact on the morphology of the nanomaterial you can have in water. So starting from the same peg, just changing the monomer can give you really 2 different kinds of elongated nanostructure. And in particular, the one made of leucine are very elongated and really anisotropic. Okay, this was the third keyword. And I will finish with the last one. That is the keyword bioactive. So it's a specific context of the bioactivity where it's even more challenging to consider the primary sequence, because basically primary sequence is at least fundamental for promoting specific interaction, host-guest interaction with any ligand, anything you can have in cells. But the question is the sequence necessary to produce all the bioactivity of proteins? And again, here, you might answer not always. And I will try to give you one of these examples supporting the not always answer. So here, the polymerization of NCA has been used to mimic the topology, but also the bioactivity of what we call antimicrobial peptide. So you are better expert than me on that, so I will not pass too long on that. But yeah, bacteria, they use this antimicrobial peptide to fight against each other. There are a lot of antimicrobial peptides known in biology, more than 4,000. You have many different structures. Many of them, they have specific secondary structures. They are amphipathic. They have, in particular, cationic and hydrophobic side chain. And this is a very, very common feature of all these antimicrobial peptides. I'm sorry for this here mistake, this typo here. Histidine is not here. But mainly the positive charge are lysine, histidine, arginine, and the hydrophobic block is this one, including methionine in some cases. Okay, so it's all about mechanism of this antimicrobial peptide. And this is something we can strongly discuss, but it is non-established that the main effect you have with bacteria, either gram-positive or gram-negative, is an effect that is named a negative Gaussian curvature effect. So it's purely a physicochemical effect where first you have the positive charge of an antimicrobial peptide that come in close contact to the negatively charged phospholipid from the cell membrane of bacteria. And then when they are in close contact, just the distribution of this positive charge, as well as the distribution of hydrophobic residue, is able to induce a membrane destabilization based, for instance, on the rigidity of the membrane and so on, that can go up to a membrane disruption. So this is something somehow very rational, and now you have people, for instance, in UCLA that are able to predict the negative Gaussian curvature effect of any peptide sequence in the genome and so on. But what is interesting is you can even reproduce this negative Gaussian curvature effect without controlling the primary sequence of peptide structure. And this is particularly interesting considering that antimicrobial peptides can be like a much simpler design if you take synthetic polymer approach, and for instance, the ring opening polymerization to make antimicrobial structure. We have faced this challenge specifically here by not directly playing with polypeptide, but polypeptoid that are like a structural analog of polypeptide, where the side chain is supported by the peptide linkage, just to introduce resistance to proteolysis. And we have made that, I will show you, to tackle specific bacteria from the digestive tract. So again, you can have N-carboxyanhydride here, so N-alkylated, that can be alkylated by a cationic charge on hydrophobic residue. And just you mix the two monomers and you make copolymer with hydrophobic and cationic charge. The good of that is it's easy to make libraries of copolymer. So you choose the size here based on the initiator. We use allylamine. And then you choose basically the hydrophobic content. And you play with the two monomers and you make mixture of copolypeptoids. Here it's example of library where we change the hydrophobic block. And we have done studies on Clostridium difficile. So this with the Institut Pasteur. So Clostridium difficile is difficult to kill using real peptide or even proteins. And this is because they are living in the digestive tract, and because of the digestion, you can degrade basically the protein or the peptides before they reach the bacteria. And now this is the main treatment you can use, the metronidazole, vancomycin, or fidaxomicin, but you have the appearance of resistance, and some of them are quite expensive. And because of that, they need new treatments. So we have, let's say, assessed all the copolymers the library I showed you against Clostridium difficile. And even so, we don't have, I would like to say, the precision of the primary sequence. As you can see here, we have a specific balance of cationic and hydrophobic residue here, where we have a good activity against Clostridium difficile. And it's quite specific because here is a lateral chain, so it's a phenylalanine-like side chain. And if you just remove the aromaticity and if you just have a cyclohexyl, you see that you fully lose this antimicrobial activity. So there is something very specific in terms of antimicrobial activity. Even so, we use a polymeric approach. What is interesting here is you can really play with the rules of this polymer chemistry to change, to adapt what we can call the pharmacophore, change the initiator, change the size of your polymer, change the nature of the cationic charge, and so on. Define what we call a selective index, what is the difference between the activity against Clostridium difficile and the cytotoxicity over a cell line that is representative of the digestive tract, so CACU2 cell. Even more interesting was the fact that it's possible, thanks to polymer chemistry, to play with the macromolecular design. So here it's an example. I will not enter in detail because you're not necessarily chemists, but just by changing the initiator, so replacing the amine by what is called LEH-MDS, so it's a strong base. Instead of to have linear polymer, we can have cyclic polymer structure. So this is the mechanism I will not stay in, but what is interesting is you can use in that polymerization process cationic and hydrophobic monomer and make amphiphilic structure, like I showed you against Clostridium difficile. So we have done that. We have made different kind of copolymerization, random or sequential. Random means that we do the polymerization directly with the two monomers, and sequential means we first polymerize one monomer and then the other one. And we have varied the hydrophobic content. And we finally achieve a specific and good activity for the random copolymerization at 50% of hydrophobic content. So this was quite a nice result, as if you compare the MIC we obtain with this structure as compared to the MIC you can obtain with an antimicrobial peptide. Even so, we are still far from some performance, in particular of the phydamoxine in terms of selective index. And we are currently working on making this selectivity better. Also, we are making in vivo study to transfer this technology to real-life application. Okay, this is my conclusion. I think I already discussed too much. Just today, it was to present you this ring opening polymerization process, like that permit to make this polypeptide, like simplified analog of proteins, and like taking just a few keywords from the life science knowledge of proteins, so the adaptation of protein, their aqueous formation, the directed assembly, and the bioactivity. And these are other keywords we are currently working in the lab, following the same direction. So perform catalysis with the system to create some sort of fluorescence that look like the fluorescence of GFP protein, to create some active membrane transport, and even to create a polymerization process allowing a sequence control of the primary sequence. And this is even more interesting. And I wish to show you that soon in another seminar. So I want to thank all the people involved in this work. Simon with all the work about composite, Chloé, all the work, and Sebastian, all the work about ROPISA process, Pierre and Bruno, all the work about the antimicrobial polymer. And all the students, and in particular the polypep team, I'm currently supervising, and with it's a real pleasure to develop all this research. And I would like to thank you for your attention. I would be pleased to answer all your questions. Y puedo hacerlo en español. Muchas gracias.
Ya, gracias. Es que tenía mi micrófono bloqueado. Entonces, bueno, le pediría a los asistentes que tengan preguntas, si quieren escribirlas en el chat o ir levantando la mano, como siempre hacemos. Yo, pues, felicidades, Colán, tu trabajo muy, muy padre y además muy bellamente ilustrado, ¿no? Y ya comentabas que, pues, uno de los retos de estos materiales es justamente la diversidad en cuanto a la composición química que pueden tener o la, ¿cómo decirlo? O sea, la secuencia, ¿no? Que pues nos mostraste polímeros con dos monómeros, copolímeros, como tú los llamas, con dos monómeros, pero no sé qué, bueno, ya ahorita nos decías que en el siguiente seminario nos lo platicarás, pero me gustaría saber cuál es la estrategia o conceptualmente cómo pensarían abordar en este tema. Esa es una pregunta. Y la segunda tiene que ver con la última diapo que nos mostrabas, de ya tratar de llevar estos péptidos antimicrobianos, que es un proyecto muy, muy interesante, ya a una aplicación. Y bueno, está bien probarlo en animales, pero me pregunto si han considerado también hacer experimentos como de ver en el microbioma pues qué efectos pudiera tener sobre esos organismos y además qué modificaciones pudieran hacerle a estos péptidos, porque como sabes, muchos fármacos se activan justamente, sufren transformaciones una vez que pasan por el intestino y se pueden absorber. Entonces esas dos preguntas.
Pues sí, como decir rápidamente sobre la primera pregunta, digamos que para mí hay dos cosas importantes en las proteínas, dos cosas. Pues, de manera muy simplificada, la primera es la secuencia, que es súper importante. Como ustedes lo saben, lo hacen con computadoras y con X-ray structure, etcétera. La secuencia sí es súper importante, pero algo que no se estudia suficiente es también que la talla sí da propiedades y es algo que nosotros en polímeros conocemos. Entonces, lo que es interesante es si alguien como yo hacemos modelos simplificados de proteína, solo con la talla es posible mimicar propiedades de las proteínas, aunque estas propiedades no son tan finas que las que pueden tener las proteínas. Eso significa que no solo la secuencia, pero también la talla sí es importante en este mundo de las proteínas. Y la talla permite, pues, hacer separación de fase, la talla permite, sí, de confinar, la talla permite, sí, de agregar, etcétera, etcétera. Y hay muchos efectos, incluido el facto que a veces solo mezclar aminoácidos sin controlar la secuencia pueden reproducir algunas actividades de las proteínas. Y yo lo encuentro muy, muy interesante. Claro que las secuencias sí son súper importantes en muchos procesos y además en los procesos que ha enseñado, las secuencias como afinan una propiedad inicial que estaba una propiedad de la talla de la proteína. Pero no solo la secuencia, pero también la talla. Ese es el mensaje fundamental, digamos. Pues en cuestión de los péptidos antimicrobial, es que pues el objetivo era de ir en el tubo digestivo y para evitar la absorción, lo que hemos pensado es que hacer una reacción de polimerización es la mejor manera de evitar la absorción de las moléculas. Y es lo que estamos haciendo. Entonces, es muy fácil para nosotros con un polímero de Polymerization Degree de 20, de pasar a un polímero que tiene 100 o 200. Y más grande va a estar la molécula, menos vamos a tener de absorción en el tubo digestivo. Entonces, permite hacer muy fácilmente un antibiótico que no va a ir en la sangre, por ejemplo. Además, hecho de aminoácidos que se va a deshacer en la naturaleza. Entonces, había muchos puntos positivos, incluido el facto que no cuesta tanto comparado a la síntesis normal de los péptidos de hacer eso. Y por eso fuimos en este proyecto. Y ahora, pues es financiado por una empresa que lo quiere desarrollar. Pues a ver hasta dónde vamos. En cuestión de citotoxicidad es muy complicado porque pues uno puede usar células, pero realmente son los experimentos in vivo que realmente enseñan algo. Entonces estamos en eso y con pues ratas y todo eso, entonces es un poquito pues complicado, toma tiempo. Pero a ver si pronto tenemos más resultados. Cuestión de ir además de pues del tubo digestivo, pues primero lo que hemos hecho con el Instituto Pasteur fue de comparar la actividad contra Clostridium Difficile a otras bacterias. Entonces sí puede haber por unas estructuras como una forma de selectividad sobre Clostridium, en particular contra las poras de Clostridium, que no es muy común, que estamos estudiando. Si no, pues hay como un interés por como lo que se llama las aplicaciones como topic, que es más como wound healing process, cosas así, que no es tan challenge, pero que puede permitir también valorizar los aminoácidos en eso, porque también para hacer péptidos es muy caro, digamos, o proteínas reales también es muy caro. Y finalmente, pues, vemos si podemos valorizar eso en otro tipo de aplicación antimicrobial, pero estamos estudiándolo ahora. No puedo decir que va a haber un futuro en eso, pero estamos intentando muchas diferentes cosas.
Muy bien, gracias. Pues no sé si hubiera otra pregunta. Creo que no hay nada en el chat y no sé si alguien le va a
No hay nada todavía en el canal de YouTube tampoco. Estamos viendo aquí en el chat, no hay. Tenemos tiempo suficiente.
Maura, adelante.
Sí, buenos días. Yo tengo una duda. ¿Qué tan susceptibles son a la degradación estos péptidos sintéticos en comparación con los antimicrobianos que no se sintetizan de esta manera?
Pues sí, es una pregunta muy, muy importante. De hecho, no sé si ya están viendo los slides. ¿Sí ven los slides aquí?
Sí, sí.
Aquí es un experimento porque son catiónicos de lo que se llama pues una digestión de tripsina, ¿no? Es un polímero aquí de puros aminoácidos que hemos hecho, mezclando lisina y fenilalanina, que sí se degrada muy rápido con la tripsina. Entonces, los polímeros es súper interesante, sí se degradan con enzimas. Pero lo que hemos hecho son los análogos en donde la cadena lateral, en lugar de estar aquí, está ubicada sobre el nitrógeno. Y en este caso no hay degradación con la enzima. Hace que, por ejemplo, si tenemos la estructura y después de la digestión de tripsina, guardamos aquí la actividad contra el Clostridium, aunque, por ejemplo, un antimicrobial peptide, que es el LL37, ya la pierde. Entonces, sí, ya tenemos esta resistencia contra la proteólisis. Pero los análogos aquí, que son aquí en alquilado, sí se deshacen por degradación de química, no de enzima, pero de química, que es una hidrólisis normal que puede ocurrir en el ambiente, ¿no? Entonces se degradan, pero después del tubo digestivo. No sé si eso responde a la pregunta.
Sí, de hecho, pues entonces creo que depende bastante, ¿no? De cómo es, pues sí, la estructura química del compuesto. Sí, muchas gracias, doctor.
¿Hay otra pregunta del doctor Agustín López?
Sí, muchas gracias, muy interesante. Me pregunto ¿qué tan cerca en la naturaleza hay estructuras como estas que están diseñando? Por un lado. Y la otra sería si existiría en el sector alimentario de los sabores alguna posibilidad de desarrollar polipéptidos con funcionalidad al sabor dulce, al sabor salado, etcétera. Si han pensado en esas opciones.
Pues voy a empezar con la segunda pregunta. No lo hemos pensado, pero sería súper interesante. Eso sería súper interesante. Pero lo hemos pensado, por ejemplo, por otras cosas como la fluorescencia, muchas propiedades de las proteínas. Para mí no necesariamente dependen de las secuencias. A veces es como cosas que se pueden reproducir. Por la primera, no sé exactamente cómo decir. Hay dos opciones. La primera sería que partes de proteínas que no tienen secuencias son difíciles de estudiar. Entonces, a lo mejor en bioquímica se dice que no hay este tipo de secuencia, pero poco a poco se ve que a veces hay, ¿no? Hay, por ejemplo, esta modificación post-traduccional que se llama poly-E, porque en química no es, en bioquímica no es poliglutámica ácida, es poly-E. Poly-E sí aparece por la ubiquitinilación, pues cuando el proteasoma quiere como destruir las proteínas, es algo en el proceso. Hay cosas aquí, a veces, hay partes de proteínas que son solo una secuencia de un aminoácido que es repetido, ¿no? Eso existe. A lo mejor no es tan estudiado porque es difícil definirlo en bioquímica, porque todos los útiles que ustedes tienen son a base de la secuencia, ¿verdad? Pero es una hipótesis, pero a lo mejor no hay tantas de eso, pero puede existir. Otra parte es también considerar que, pues, al origen de la vida no había secuencia, ¿verdad? Entonces, ¿cómo fueran construidas estas proteínas? Y es algo que encuentro muy interesante. Es como definir cuál fue como la fuerza motriz de todo eso. Muchas personas piensan que desde el principio había que definir una secuencia perfecta para tener una actividad de algo. Yo creo que no, y yo creo que realmente lo que fue importante al origen fue una talla para tener pues un compartimiento, para tener como un efecto de segregación, de agregación, etc. Y son procesos simples, poco a poco, que se han afinados con la secuencia. Y entonces había como un mundo con menos secuencia que ha evolucionado con más secuencia, digamos. Pero la química de los polímeros nos permiten ver más eso, digamos.
Ya, muchas gracias.
Te tengo una pregunta, si me permites, Mar.
Adelante, adelante.
Gracias, gracias. Pues interesante, muy interesante con la plática. Y mi pregunta va más orientada hacia el tema del campo de ingeniería de tejidos, porque uno de los materiales que se utilizan es este polipéptido, el poliglutámico, dentro de esa área, ¿no? Como soportes y demás para el desarrollo de diferentes modelos celulares. Más allá de este soporte, que digamos es el más usado, hay otro tipo de modelos de estos que se pueden diseñar con proteínas para ese tipo de propósitos, Alan.
Sí, entonces, por ejemplo, yo tengo algo en la memoria, que aquí se puede hacer una copolimerización de 3 monómeros, que sería el R, el G y el D. Y entonces, de mezclar los 3, da polímeros con esta secuencia de 3 aminoácidos que se supone importante para tener esta adhesión de las células, ¿no? Para hacerlas crecer en ingeniería tisular. Pues, si se mezclan los 3 aminoácidos aquí, como de manera estocástica, digamos, si alguien hace un polímero de 100 aminoácidos, él va a tener como, creo que es 6% de la secuencia que va a estar este tripeptide, pero solo al azar, ¿no? Digamos. Entonces, usando estos polímeros va a promover esta secuencia de 3 aminoácidos que permite a las células de tener adhesión, ¿no? Pero a muy bajo costo. Eso fue publicado, por ejemplo.
Ok.
Entonces, con consecuencia simple de algunos aminoácidos, sí, mezclar los aminoácidos, sí, es muy, muy interesante.
Claro, claro, sobre todo por esta naturaleza, digamos, mucho más compatible, ¿no? Con las células humanas o que se desea crecer en este tipo de soportes, ¿no? Creo que hay
Sí,
una gran ventaja, ¿no? Y digo, más allá de las cuestiones de compatibilidad, quizá de estos temas que tienen que ver con ángulo de contacto, hidrofobicidad, etcétera, ¿no? Que en el campo de materiales es fundamental, ¿no? Así que...
sí, además que si se considera, por ejemplo, usar directamente las proteínas, a veces es difícil como conseguir como 1 gramo de proteína, ¿verdad? Porque es difícil como producirlas. Aquí con polímeros así, una reacción puede, pues, producir fácilmente 100 gramos, 1 kilo de polipéptidos. Entonces, realmente las aplicaciones posibles son diferentes, digamos. Complementa esta manera de hacer los polipéptidos otras aplicaciones, y permite como lograr aplicaciones que no fue posible antes con los péptidos o las proteínas reales, con realmente una secuencia, digamos.
Okay. Gracias, Colin. Gracias, Marco.
Gracias. Hay una pregunta en la gente que está viendo YouTube, Alejandra Valdés. Dice, hola, soy Alejandra Valdés, estudiante de biomembranas del CIMBESTAB. ¿Han intentado acoplar estos bioconjugados en modelos de liposomas? Y si tienen resultados previos, supongo preliminares, sobre la citotoxicidad en modelos de ratones. Entonces son dos preguntas.
Pues lo que se hace es que se construye lo que se llama Polymersome. Es como un equivalente de los liposomas, pero hechos de polímeros, hechos de estos polímeros de aminoácidos. Y sí es algo que es muy desarrollado ahora, como desde hace 20 años. Entonces, la llave de eso es construir un polímero que tiene dos bloques. Un bloque que es hidrofílico y un bloque que es hidrofóbico. Con autoasamblaje sí es posible tener como una vesícula, que se llama un polimersome. Y sí hay muchos ejemplos ahora de estos polimersome que son compatibles, que se usan en nanomedicina, y sí tienen ventajas comparados a los liposomes. Y por ejemplo, aquí de lo que he enseñado, el stimuli responsiveness. Entonces son estructuras que se pueden hacer o deshacer con respuesta a un estímulo, por ejemplo, que puede ser como he enseñado, un pH change o la presencia de algo en el medio, etc. Pero sí hay muchos estudios en este campo.
Y creo que te preguntaba también si tienen resultados preliminares sobre qué tan citotóxicos en ratones son, bueno, tóxicos en ratones son tus péptidos antimicrobianos.
Sí, pues por el momento, pues eso es muy preliminario y eso es como la cantidad que damos de comer, ¿no? De los polímeros. Hay que ver que son finalmente polímeros que son como carne, ¿no? Porque son aminoácidos, son proteínas. No son tan tóxicas aquí, por ejemplo, pero eso es como un estudio que hemos hecho hace más de un año, entonces no es muy representativo, ¿no? Parecen que pierden un poquito de peso, que luego lo toman de nuevo, hay algo así que es un poquito chistoso, pero aparentemente hasta 75 miligramos ya sobreviven, que es bastante alto, entonces comen eso básicamente, que es bastante. Entonces no es tan tóxico por el momento, pero no puedo dar más estudios en este campo por el momento, pero va a venir muy pronto. Estamos sobre el camino, digamos.
Muy bien, pues muchas gracias. Si ya no hubiera más preguntas, pues te agradecemos de nuevo, Colán. Muchas gracias por este seminario tan interesante y nos vemos pronto.
Sí,